Good afternoon. I believe we'll go ahead and get started. It's a pleasure. My name is Warner Greene. I'm the director of the Center for HIV Cure Research here at the Gladstone Institutes. It's a pleasure to invite you to this briefing on the 2019 novel coronavirus outbreak. I am joined today by two distinguished scientists, Dr. Charles Chiu, who is a professor of medicine and laboratory medicine at UCSF and also the director of the UCSF-Abbott Viral Diagnostics and Discovery Center. And also my close colleague, Dr. Melanie Ott, who is a senior investigator here at the Gladstone Institutes and professor of medicine at UCSF. Both have wide-ranging expertise in virology, and I hope that we have a very robust discussion this afternoon. So what we will do is to begin just with a bit of background. All speakers have promised that they will not take any longer than 5 minutes. So we should be within 15 minutes into the panel discussion, and at which time we'll be able to answer your questions. I would remind you that we are live streaming. So we want to make sure that if you have a question to ask, that you do so with the microphone so that the live streaming is effective. So why don't we begin? So I will start with what I call coronaviruses 101. So what is a coronavirus? Well, it's actually a family of viruses, RNA viruses, and these are large viruses. That accounts for 10 to 30% of the common colds that we have in adults. Rhinovirus is, of course, the most common cause of the cold, but 10 to 30% of colds are caused by one form or another of a coronavirus. But there can be serious outbreaks of coronavirus infection. Different strains of the virus can cause these outbreaks. For example, as you all are familiar with SARS in 2002, the severe acute respiratory syndrome broke out. We see, in fact, that there was, at this point in time, there were 8,098 individuals who were infected that were reported, that we know of, and 774 deaths. In 2012, there was another coronavirus outbreak called the Middle Eastern Respiratory Syndrome, or MERS. Here there were 2,494 infections to date, and 858 deaths. So you can see that with SARS that the death rate was about 9%. With MERS, it's up around over 30%. So where do we stand now with the latest outbreak, the 2019 novel coronavirus? So in fact, the stats, and nothing is certain at this point. We're in the midst of this outbreak. Currently, though, we have, as of this morning, 28,353 infections, 565 people have died, and now the virus has been reported in 28 countries. Certainly, the epicenter of this outbreak is in China, and the true epicenter is in Wuhan in the Hubei province. So where do these coronaviruses come from? Well, in fact, we know that the primary animal reservoir for these coronaviruses is bats. But generally, the virus does not transmit directly from a bat to a human. It goes through a secondary host. And for example, in the case of SARS, it was the palm civet. And in the case of MERS, it was dromedary camels were the primary conduit. Now, we don't know what the animal reservoir was for the novel coronavirus. We know that the outbreak appeared in a seafood market in Wuhan, but we still don't know what that intermediary host was or is. Now, it's important to note that these viruses use specific cellular receptors to get into cells, and the cells that they're primarily infecting are cells in your lung, ciliated epithelium, pneumocytes. They also can infect enterocytes in your GI tract that can lead to diarrhea. But the principal manifestation is a severe acute respiratory infection. And SARS uses the angiotensin converting enzyme 2. I think Melanie will say more about this. MERS used a different surface peptidase, the dipeptidyl peptidase 4. And the early studies suggest that the angiotensin converting enzyme 2 is also used by the novel coronavirus of 2019. So in this slide, I try and depict, just give you a sense of what's going on. This is originally this was sourced from the New York Times. So essentially, I've listed here several viruses. Viruses that, for example, bird flu, Ebola, MERS. You see, in fact, on the y-axis is the fatality rate, and on the x-axis is the number of transmissions per infection. So what the number of transmissions per infection is what we call R0. And in the case of, for example, measles, if you're infected with the measles, it's likely that you will spread that virus to 15 other individuals. With chickenpox, it's like 8.5 individuals. Now, in the case of the common cold, we're down around 2. Now, these viruses are not very lethal, but as you move up the y-axis, you see Spanish flu, MERS, SARS, Ebola, bird flu. These are the big killers. But you also see that their transmission is lower than that per person than chickenpox and measles. Now, we're not quite sure. So there's a big box here for the 2019 coronavirus. It looks to me like it will probably have an infected individual will likely spread the virus to 2 to 3 additional individuals. Now, it could be bigger than this because there may not be a reporting of all of the infections. It also looks like that the mortality rate is around 2%, but again, we don't know for sure what the denominator is. Well, there may be a lot of undiagnosed infections, and so the death rate or the mortality rate could be smaller. So there's a lot of uncertainty, and that's why there's this big box. But my sense is that this virus looks like it's a little bit more infectious than SARS and a little less lethal than SARS. So just, but take that with a grain of salt because the data are rapidly evolving. That said, it's an important perspective that there's so much concern about this coronavirus outbreak. But indeed, this is not the biggest threat that we face day to day. The biggest threat that we face from an infectious disease RNA virus is influenza. And we're in the midst now of the flu season. There have been, here in the United States, there have been 19 million infections. There have been 180,000 hospitalizations and 10,000 deaths. And we're not through this flu season at all. In general, around 35,000 people die every year of the seasonal flu. So I'm sure you all got your flu vaccines. This year's vaccine is not perfectly matched, but it is matched with 2 of the circulating strains. And if you were infected with one of those strains, it could save your life. So the important point is that not to be overly alarmed by the coronavirus. I mean, I think we should follow with interest. Follow what's happening. But this is, there are lots of reactions that are occurring. For example, the Chinese government has quarantined major large cities, an unprecedented level of quarantine. In general, if you look at the history of quarantines, they often occur too late, and they are actually disruptive themselves. I think we now see or hearing reports that in Wuhan that there are sick individuals who are wandering from hospital to hospital trying to get care. They're running out of supplies. The medical systems is completely stressed. You've seen the remarkable ability, the remarkable building of new hospitals within days. So there's a lot of stuff going on. There's a lot of policy. The United States' own quarantine rules have been put in place. I'm sure we can talk more about that. But my message is that this is an outbreak. It's not an epidemic. We want to keep aware of this particular virus, but it is not the biggest threat that we face day to day in our lives. Certainly not here in the United States with only, I think, 12 infections documented. So with that, it's a pleasure to introduce Charles Chiu for his remarks. Again, Charles is a professor of medicine and laboratory medicine at UCSF.
Thank you. Okay, great. Good afternoon. I'd just like to start by thanking the Gladstone Institutes and Dr. Warner Greene for inviting me to speak with you today. I'll be going over some of the epidemiology of and a timeline of the outbreak, and I'll also talk just one slide about what we're currently doing here at UCSF. So this is a summary. One thing is I had to be very selective because I noted that there were already over 15 publications regarding this novel coronavirus in journals over the past week alone. I've focused on just a few of the publications. There's one in the Lancet which goes over a timeline of the outbreak. So I just want to briefly review it with you. It was first reported a cluster of cases of pneumonia that were basically seen of unknown origin was reported to the China National Health Commission in December 30th of 2019. By January 1st, the Huanan seafood market, which was thought to be the origin of the virus, was closed. The novel coronavirus was actually isolated roughly just 1 week later. And I should also mention that Chinese scientists have actually made significant progress. They were able to, for instance, make public the genome of the virus in just a few days after identifying it as a novel coronavirus. They've also generated already diagnostic tests, nucleic acid-based diagnostic tests, as well as antibodies. And also, reportedly, there's an animal model as well that has already been developed by Chinese scientists. The first fatal case was reported in January 11th, and in January 13th was the first case that was reported in Thailand. The identity of the virus, the official name of it, was in January 12th, where it was named officially as 2019 nCoV, or 2019 novel coronavirus. And then you have scattered cases elsewhere in Asia as well. And then as of the last week of January, then there were the total number, hundreds of cases that were being reported, mainly from Hubei province, but mostly from Wuhan city. I also show you a slide basically on the right side, where this is published last week in the New England Journal of Medicine about the first 425 confirmed cases in China. And it gives you some idea of the days from infection to symptom onset. So that was a big question. What is the actual incubation period of the virus? And it's thought that, in general, it's very similar to other coronaviruses. It's thought to be mainly from 2 to 14 days or 2 to 11 days, if you want to take the peak of it. Appears to be about at around 4 to 5 days, and that is consistent with other coronaviruses, including SARS and MERS. Similarly, there is also another timeline you see here, which is days from illness onset to hospitalization. And I think this is really important because the days from infection to symptoms may only be on a median of 4 to 5 days, but then you have additional days before a patient actually develops severe enough symptoms to seek medical care and be admitted to the hospital. And that happens to be, on average, it appears to be a median of approximately a week or two, a little more than a week. This is basically a slide that shows you, I got this last night, so it may not be as updated as Dr. Greene's, but you see here that we have a total of 28,000, roughly 20,000, it's probably over 30,000 at the time confirmed infections, 12 cases to date in the United States. The most latest was in Wisconsin. This is actually provided by an online resource provided by the Johns Hopkins GISAND, so you can see that. I also want to briefly go over the genomic epidemiology. There are a lot of questions about this virus. Has this virus mutated? Is it going to mutate? What's interesting is that so far, as of yesterday, there have been 60 genome sequences that have been deposited and made publicly available. The phylogeny, so what you see here is a phylogenetic tree. It shows the evolutionary relationships among the different coronaviruses from different patients. Notably, all of these genomes are highly related. There are at most 7 mutations relative to a common ancestor. And if you do molecular clock analysis and you track it back to the date of the common ancestor, namely the initial animal to human transmission event, it probably occurred in late November, early December. And since then, there's been no evidence of ongoing animal to human transmission events. All of the outbreak has essentially been sustained by human to human transmission. And then this is the second to last slide. This is a description of the first case of the 2019 novel coronavirus that was reported in the United States. This was the case that was reported from Washington State. A couple of things of note are that you can see here based on the symptom map that fever, cough, and fatigue are really the most prominent and persistent symptoms, especially when a patient begins to feel ill. Pneumonia develops typically later. It happens, at least for this patient, it happened 3 to 5 days after the patient was admitted. And if you recall the average time between the incubation period of, let's say, 5 days, we're talking that severe disease may not develop until 7 to 10 days after a patient initially becomes infected. Interesting also, the one other thing I want to note is that the virus has been detectable in respiratory secretions for up to 12 days after the onset of illness. And even at day 7, there were moderate to high levels that were seen. Notably, for this particular patient, this patient only began decompensating by days 6 to 7. On day 4, the patient simply had mild respiratory symptoms, but during that period, that patient actually had the highest level measured of viral load. And this is of significance with respect to transmissibility. And this is the last slide I have. We are developing a test for rapid detection of 2019 coronavirus infection. The CDC yesterday received EUA approval, emergency use authorization approval from the FDA to test for the virus. They have an RT-PCR assay that uses 3 targets to test for the virus. This is an assay that's being rapidly deployed to both state and local public health departments. We're developing essentially a point of care assay that we feel might be complementary, and it's our hope that we can develop this. We're developing this in collaboration with Mammoth Biosciences, and we're using a CRISPR-Cas-based approach to be able to diagnose the virus within 1 hour of collection of sample. And that's the last slide, and I'd like to introduce Dr. Melanie Ott, who will be talking more about the virus itself. Thank you.
Well, thank you, Charles. Good afternoon. Let me just start by reiterating that what the previous speakers have already said, that this is a very scary and fast-moving infection, but it seems by pure numbers to be much less dangerous than its cousins, the SARS or the MERS virus. And this is shown here in the numbers from the American Society of Microbiology. Numbers, as you see, are outdated by the day because they increase by the hour, but also by this larger study from clinical features of patients hospitalized in China. And so the question is now, is this really a less lethal virus, or are we taking better care of these patients? And it's probably a combination of both. You might notice here on the bottom of the slide that from the 41 hospitalized patients in Wuhan described in the Lancet, 38 were put on antiviral therapy. So what is this antiviral therapy? Just to be clear, there's really currently no specific therapy for the novel coronavirus. All we're doing is borrowing from other viruses. And we're borrowing from the flu virus, we're borrowing from HIV, and we're borrowing from Ebola mainly. So all patients in Wuhan have been treated with Tamiflu. That's probably very familiar to many of you, that you know if you take early enough in a flu infection, it might have some activity in shortening the symptoms. That is a drug that is relatively specific to an enzyme in the flu virus. But the Thai colleagues in Thailand have reported yesterday that if you use it in combination with the HIV drugs at relatively high doses, they seem to see an effect. The other drugs are the HIV drugs. These are protease inhibitors, extremely effective against HIV, but also in the past having been tested against coronavirus because coronavirus is expressing two forms of a protease. So currently, it is probably the most recommended drug in combination with interferon alpha, which is a relatively unspecific antiviral drug that we give often in viruses where we don't have another choice. Currently, it's mainly also hepatitis B infection, but hepatitis C was also treated with interferon for a long time. And so the last drug that is currently rising is a drug that is currently not FDA approved. Both the other ones are available off the shelf and are orally also bioavailable. Remdesivir, which is a drug that has been developed against the polymerase of the Ebola virus, is currently in clinical trials. This was used in the first US patient that actually Charles showed you the data for. And showed some efficacy, but all of these drugs have not been tested in any standardized clinical trial, so we have no idea whether what we're doing is effective or whether it is just the natural course of the infection in that specific virus, in that specific patient. Interestingly, Gilead announced yesterday that because of the promising results with its drug, they're starting a phase 3 clinical trial in China now, which will give us a lot of answers that we currently don't have. So these are the repurposed drugs, but there are drugs in the pipeline currently. Let me start that interest in making a vaccine is very high currently, and there's at least 3 companies that have publicly announced that they are going after the novel coronavirus, either with a DNA, an RNA, or with a nanoparticle vaccine. But the other possibility is to really go very actively using isolated broadly neutralizing antibodies that have been very promising against many viral diseases in the past, most importantly Ebola in the last few months, where it showed really extremely good effectiveness when it was administered early in the infection. So Regeneron has a broadly neutralizing antibodies against MERS already produced, and this is currently already in clinical trials with 48 patients against the novel coronavirus. And also Vir here in Mission Bay is currently evaluating safety and efficacy of its previously identified anti-coronavirus monoclonal antibodies, which is not specific against this coronavirus. And it will see if it has any efficacy against the 2019 novel coronavirus. There's also a lot of noise about another polymerase inhibitor that is similar to what Gilead had produced with its drug. It's way further down in the clinical development at this point, but it has very broad effectiveness. It's also a polymerase inhibitor and has very broad effectiveness against 20 RNA viruses, including coronaviruses, and it is considered as a potential promising drug target in the future. So this is what is sort of coming up, what is in the pipeline. The future opportunities, I want to go back to what Warner explained to you at the beginning about the receptor. I think many people in the past have focused on, the SARS ACE2 interaction. So ACE2 is the angiotensin converting enzyme 2 that has been identified as the receptor for the SARS virus. 2 publications came out a few days ago indicating that this is very likely also the receptor for the novel coronavirus. This offers unique opportunities for future therapeutics that are outside the regular small molecule or even antibody realm. I think people have, thought about using parts of the ACE receptor as a decoy to neutralize the virus, or vice versa, use the binding site in the spike protein of the coronavirus to sort of decoy the ACE receptor. There's also ideas to using antibodies against or inhibitors against the ACE2 receptor, which have been developed in the past against the SARS virus. So there is a lot of possibilities here that are currently mainly centered around this receptor, but it will very much depend on whether this is the only receptor or whether it's the true receptor. Also, everything is currently speaking for it being the real receptor. So what are we doing here at Gladstone? I think we're doing quite a bit. I think we are also involved in a similar development of a diagnostic test that Charles has described to you, but with a different CRISPR enzyme. We're also having an active flu research currently ongoing, where we're using primary human organoid models to study the virus, which gives us a lot of opportunity to use this for coronavirus in the future. But in general, we also have a long-standing interest in developing broad-acting, host-directed antivirals in our biofulcrum program here. What we're doing here is really leveraging basic science that's ongoing at UCSF, Gladstone, and at Berkeley in multiple areas of proteomics and CRISPR biology to hone into certain cellular pathways in the cell that are commonly used by multiple viruses. So we're not really so much focused on one individual virus, but more on groups of viruses or viruses even in combination with other pathogens like bacteria, because the hope is that we will develop drugs against these pathways and then be able to better capture or be effective against emerging viruses like now the novel coronaviruses, because we're not so highly specialized on one single virus. So with this, I end and invite my colleagues up to the stage for the discussion part of the program.
So the floor is now open for any questions that you might have. Again, let's use the microphone because we are live streaming. Yes.
Is this on? Yeah. So you pointed out clearly this is not the same kind of risk that we run with influenza. But what are the events that happen when a virus goes from being an outbreak and then contained to something that spreads wide enough to where it is with us forever? Because if this were something like the flu, where we're facing it every year, clearly the death rate is higher. It would be a significant difference.
So the cold, the coronaviruses that cause cold kind of wax and wane with the climate, like influenza. There's a flu season and not. But these serious coronaviruses don't have that same type of seasonality. SARS was not controlled by medicines or a vaccine. It was controlled by public health interventions, tracking infections, limiting contact of infected individuals with others, and certainly in the absence of appropriate vaccines and highly active drugs, that's exactly what will be undertaken here. As you can see, I was kind of referring to the quarantine of a city of 11 million and other cities in the province. But you see, in fact, that the spread has now essentially involved every province of China. So, as is often the case with these types of, and this is unprecedented in terms of scope, they're often too late, and they wind up causing a lot of, for individuals who are quarantined, it's very, very difficult. Other questions? Oh, come on.
Can you hear me? Okay, sorry. Thank you for this information. I have two questions in terms of the timelines of publications of all these papers that have come out in such a period of time. What about the peer review process? What's coming out? What you can trust, not trust? And the other question is pertaining in what's happening with children. There's been only a very few cases reported in children, and is it because we don't have the numbers yet, or is there something specific to kids' immunity which makes them more resilient to the virus?
Yeah, so I can perhaps address the first question, which is a timeline for publications. I do know that all of the major journals have essentially expedited the papers or manuscripts that relate to the novel coronavirus. So certainly peer review would be expedited. Many researchers have gone ahead and deposited their preprints into bioRxiv as well. So I would encourage you, if you're interested in remaining in reviewing the preprints as well, although those are not officially peer reviewed yet. In addition to that, I think that with regards to the reliability of the published data, I think it's really important to realize that this is a very rapidly changing outbreak, and as a result, even publications in perhaps what's regarded as the most well-respected journals, the authors may, but there was recently, for instance, an erratum that was issued for a New England Journal of Medicine paper that described the German cluster where they described asymptomatic transmission. It turns out that it wasn't really asymptomatic transmission, it was minimally symptomatic, and they had actually gotten the information secondhand, and they had never directly asked the patient whether or not she actually had symptoms. So I think the justification for that is the importance of actually getting this information out to the public as soon as possible. One option would be, if you're interested and you're working in this area, is actually to release your data as a preprint. And many journals will accept preprints and still be able to expedite the peer review process.
I agree with this, but I just wanted to say one thing. I think you pointed out very elegantly that the symptoms at the beginning are very unspecific, and the pneumonia and the respiratory symptoms really start very late. So it's really hard to say who is non-symptomatic and who is symptomatic in that case. I also want to say that there is a word of caution. There is also quite a bit of interesting stuff circulating, not so much in the peer-reviewed, but in other medias around, including that the coronavirus diverted from HIV and other things. These are not been in any way have a scientific basis.
So this New England Journal article was very important because it suggested that people might be shedding virus before they had any symptoms. And so you can see that the screening in the airports being used for fever, that, you know, if people were actually shedding virus before they got sick, that wouldn't be useful, really all that useful. So the New England Journal, the first patient that was thought to be passing it in an asymptomatic way, as was said, actually did have symptoms. But there's another patient that acquired it from that one who then passed it to a second patient, and that individual was not did so before there were symptoms, and was never exposed, and the other individuals were never exposed to the first one. So it's I'd say it's a we don't know yet. We don't know yet. But if it's like other coronaviruses, it's very likely that it will be passed once you develop symptoms. Also, with the coronaviruses, for example, with MERS and with SARS, that in fact viral load increases. It's delayed, increases at 6 to 8 days after symptoms, and that puts the medical community. There is a tremendous number of nosocomial infections with these coronaviruses because it's the physicians and the nurses that are caring for these people in the hospital when in fact they are shedding virus at the highest rates. There are the classic unbelievable outbreaks of MERS in hospitals that spread to 186 people. It's a wonderful epidemiological study, but the delayed production of virus puts the medical professionals at particular risk with this virus.
I think the children question is very interesting. I don't know. I think we have to wait and see. I don't know if there's anything particularly known with the SARS or MERS virus with children that stands out, but maybe.
I mean, there is some data from the SARS and the MERS literature and the reporting that children, for some reason as well, they tend to, even if they're infected with SARS coronavirus or MERS, as well as this novel coronavirus, they tend to have fewer symptoms and are less likely to deteriorate clinically. So the basis for that is really unknown. But this virus, the 2019 novel coronavirus, appears to fit the same pattern as SARS and MERS in that folks who are, or patients who are elderly or who may have pre-existing conditions, heart disease, diabetes, liver disease, et cetera, are more likely to decompensate and develop pneumonia and life-threatening complications as a result of the infection.
Elderly defined as older than 60. Bob.
I'm interpreting what Dr. Ott had to say as there is no evidence to support that sort of tabloid thing we've been hearing, that this came out of some lab someplace, a mistake. But as a layman, I'm wondering, for years we've been hearing warnings about overuse of antiviral medications and things like that. Could that be something that contributes to the mutations you've seen as opposed to sort of a natural pattern of mutation?
No, I don't think that, this is a virus, this is a zoonotic infection. It starts in an animal, or in a bat, and likely passes to another animal and then into humans. And then the virus has adapted in that intermediary host to improve its ability to spread human to human. And that's what we're seeing now with this outbreak. So I don't think that we don't have antivirals. Maybe we would see the emergence of antiviral resistance with the passage of time. But no, this is not something that was man-made in any way. We are simply the end host of this chain of infection.
And the virus in general is not regarded as a very fast mutating virus. It is unlike other viruses that we have who can adapt very fast. So I think also the accumulation of mutations in that outbreak, I think, indicates that it's probably not going to be mutating at a very fast rate.
So this just has to do with sort of the attempt to contain this. It seems like China is doing kind of Herculean efforts to building a hospital of 1,000 and so on in 10 days and so on. But what worries me is that once this gets into India and other countries that are much less able, for instance, to pull off such a containment, that it will be very difficult to contain. What can you just comment on that?
Yeah, it's a great question. I think the first thing I would like to stress is that there's no evidence of sustained transmission in any other country other than China. So essentially, all of the cases that have been reported so far in other countries have really been either returning travelers from China, and or their close household contacts or healthcare workers. And I completely agree with you. I mean, I think right now we're actually currently at a transition point, because the question is whether or not the outbreak can be contained in China. If it cannot be contained in China, if these measures that and if enough resources are not really devoted towards addressing the outbreak in China, it's only a matter of time, I believe, before you will see infections spill over. And it really doesn't take, as you can see, that much undiagnosed infection for it to, say, lay hold in a different country or become established in a different country. I agree with you that many other countries, especially in the developing world, are really poorly equipped to be able to manage this magnitude of an outbreak. We already see some examples, including, for instance, the Ebola virus outbreaks in West Africa, just the difficulty in low-resource settings of being able to implement even basic public health interventions to prevent spread. Notably, in Africa currently, there are actually only 2 laboratories in Africa, one in South Africa and one in Senegal, and these are both large national reference laboratories that can even test for the virus. So that gives you some idea of kind of the magnitude of the problem that would occur if this virus is able to exit China and basically become established in another country. I do think, though, that there have been efforts, for instance, the Bill and Melinda Gates Foundation have dedicated $100 million to helping to fight and contain this viral outbreak, of which a substantial portion will be bolstering up low-income, low-resource countries and their public health infrastructure. So if there does happen to be a local outbreak, it has a better chance of being contained.
So part of the etiology that's been described for some of the patients that go through this decompression and also degrading health status was cytokine storm. So that indicates a very robust response from the host. Are there any therapies that are being tried to help those people from the host perspective?
Do you want to comment? You can comment.
Well, I mean, it's a very interesting question. I think this is something that is currently been studied, and the indication is that there is, very specific cytokine induction in different cell types, and that these can, lead to a cytokine storm, sepsis type of deterioration of the host, and I think everything that we know about neutralizing these cytokines is going to be applied to interfere with the acute situation in these patients.
Yeah, I would just like to comment that it's actually it's currently very difficult to study these questions because unfortunately, cultures of isolates, really unique cultures of the virus to be able to study this both in cell culture, organoid models, or even in animal models. But one issue is that we don't even understand, for instance, with respect to the lung pathogenesis, whether the much of the pathology, the pathophysiology is due to direct invasion or damage of the lung, or is it due to an autoimmune response or a cytokine storm? So these fundamental questions really need to be studied, and much of the focus has really been, understandably, on the epidemiology with respect to the research being done and to aid the public health response. But I think ultimately we'll need to do the basic science to really understand that, to be able to really address, develop targeted therapies that would be effective.
Thanks for the information, guys. So I had two questions. One is, what's the major route of transmission for the virus? I noticed that there were positive PCR detection tests in fecal samples, and I wonder if that's a major contributor of viral spread. My second question is, do patients that are infected and recover develop a sterile immunity to it, or is there a possibility of reinfection?
Well, I will talk about spread. In general, it's respiratory droplet spread, but there can be fecal-oral spread as well. But respiratory droplet is the principal. That's hand washing is critical to kind of interrupt that chain.
And the question is whether or not there's a sterile immunity to it. So the answer is we don't know. We do know that there are 4 circulating human coronaviruses, OC43, NL63, HKU1, and 229E. So these are essentially seasonal human coronaviruses that, as Dr. Greene said, are thought to cause anywhere from 10 to 30% of all respiratory infections. At least with those coronaviruses, it does appear that if you are infected, there is a period of time where that you develop some immunity to it. Now, to that particular strain, I should stress. And we don't know whether there's cross-immunity if you get infected by a different strain of coronavirus. And we also don't know about the durability of that immunity. It certainly is longer than the immunity that you get, for instance, from influenza. But certainly over time, the antibody titers will wane, and presumably you would lose immunity. It's very difficult to extrapolate, though, with respect to this virus, simply because it's so new, and we don't know yet, for instance, whether patients who have recovered, are they now immune, or do they have risk of perhaps contracting the disease again and having a milder illness? We simply don't have answers to those questions yet.
Well, this is a very important question for the vaccine. Because if patients don't really develop sterilizing immunity when they have the infection, then a vaccine is going to be having very poor chances. So I think these questions will all be, addressed when we have better models and also when we have potentially first vaccine candidates.
It's interesting that there was a SARS vaccine developed, but it wasn't tested because the SARS had receded by the time it was available. There was interest in the SARS outbreak in terms of using convalescent serum or plasma. But again, you know, in an uncontrolled kind of anecdotal way.
It seems to me that the if a vaccine were going to be tried, that to scale up rapidly, that an RNA or a DNA approach could be, at least theoretically, faster than a protein-based one. I've just lost track. Has that been done on a large scale, DNA or RNA vaccines? And also, have there been, the other question was, are there vaccines against the other circulating coronaviruses?
So the current attempts are including DNA and RNA in vaccines and approaches, and I think that has been tested, I think, against MERS also in terms of a development, but not yet in a final product. I think the nanoparticle approach was particularly interesting because it is one that was just also approved for the influenza vaccine, and so they have a very good platform to go into there. But I think in terms of fast response, you're totally right. I think DNA and RNA vaccines are probably faster to adapt to that specific virus.
In addition to development in companies, which is primarily where vaccine development is occurring, the NIH or NIAID's Vaccine Research Center has launched a major effort. Tony Fauci is suggesting that they will have a vaccine candidate within about 3 months, but that it would take probably about a year for safety and validation.
And the NIH is very closely working with all these candidates because there's really a concerted effort to, include every available avenue to work together to be fast. And that goes to what Charles says. Right now, we are all a little bit sitting on our hands because we cannot get our hands onto the virus because there's no distribution of any viral isolates or even samples. And so I think there is really a need to accelerate that so that many more people can work on it and we can move faster on the basic biology side. Right now we are sort of working off the genome sequence of the virus basically in large extent.
Any other questions? Okay, well,
does a mask help or hurt?
So CDC does not recommend wearing masks. Clearly, medical personnel in close contact caring for patients with the coronavirus should take all precautions. But in terms of day-to-day interactions, hand washing is far more effective. The surgical mask, it's not protecting you from acquiring a virus from someone else. What it's designed to do is to keep the surgeon's germs, if you will, out of the operating field. But it's not designed. So the N95 masks are better in terms of containing, protecting you, but often those are ill-fitting, and so the virus just goes around the corner.
Yes, so the one exception is that, so the recommendations by the CDC are for healthcare workers if they're treating suspected or confirmed cases of the 2019 novel coronavirus to wear masks. And typically we would use N95 respirator masks that have been specially fitted for the healthcare worker. In addition to that, the recommendations are to put a mask on the suspected patient or individual who may be infected with the novel coronavirus as well, but not to wear masks in public. And it's also recommended that that members of close household members or contacts of a known or suspected infected case should also wear masks. But aside from that, there are no recommendations to wear a mask in public, at least not the current time in the United States.
So I have a question about the race and ethnicity, about the transmission of the coronavirus. Do you think there is a different rate of transmission or mortality in different races or ethnicity? Or have you already seen this in other types of coronavirus?
So essentially all of the coronaviruses, just like influenza strains, they start in Asia. And probably because of the very close interplay of humans with livestock, with other animals, et cetera, so there's an admixing of these viruses. So, for example, when the CDC tries to predict what flu vaccine to make, they're sampling in Asia, they're sampling in China, trying to figure out what viruses are circulating there to try and make the best flu vaccine possible. And so it's not, it's not unusual that the three coronavirus, or two of the three coronavirus outbreaks have kind of started in Asia, in China. It's nothing to do with the Chinese as a people. I think it has to do with their close association and interaction with animals, et cetera, propelling this type of zoonotic infection.
Yeah, perhaps also to add on that, there currently is no evidence that ethnicity influences either the severity of the illness or how transmissible an individual is when infected.
What is it there anything known about these coronaviruses actually in bats? Like, what's their physiology and what's going on in the primary reservoir?
So yes, bats are the primary reservoir. There's a slew of these coronaviruses in the bats. The bats aren't sick. They're not dying of coronavirus respiratory distress, but they are. And there seems to be this unique secondary host. Like originally when SARS, it was the civet, the cat. But then it was subsequently figured out that in fact the civet got the virus from the bats. And there's a whole slew of these coronaviruses in bats. So maybe the next coronavirus, maybe we ought to be looking at bats more carefully.
I think it's being done. I think there's systematic sequencing of viruses in the bat, and they have, I think, discovered up to 60, but they're continuing. They know that it's not, the end of it. And there's also interesting first biology been done to show that the bats actually don't recognize these coronaviruses in the same way like we do, and that's why they have less inflammation and less, disease development. So there's beginning really interesting studies coming out there to show that these bats have, because of unique, innate differences with us, an advantage on hosting and developing these viruses.
Yeah, one last thing is that based on the bat coronaviruses that have already been sequenced, actually this virus, this novel coronavirus, actually has 90 to 94% identity to bat coronaviruses that have already been sequenced and were placed and added to the database. This virus, in comparison, only has 80% identity to SARS coronavirus, which is its closest relative among the human coronaviruses.
Well, I would like to thank you for attending this briefing, and for those of you online, thank you for listening. And I think we'll bring this to a close. Again, the final message I think we want is everyone should be watchful. We should follow this outbreak, but we should be secure in the fact that the apocalypse is not upon us. This is a viral outbreak that is that's running its course now. And there's a lot of development in terms of therapeutics and vaccines. We're getting better at dealing with these viral infections. I mean, as Melanie mentioned, tremendous progress with Ebola. So I anticipate that we're going to get better and better at handling these coronavirus infections in the future. But right now, it's not here in the United States, as we've talked about, only a dozen individuals infected. I wouldn't be surprised, of course, I would predict that there will be more cases. There'll be more cases as slow and steady spread, but using public health measures until we have medicines and vaccines will control this coronavirus infection. That's my prediction. Thank you, everyone.